Good morning, good afternoon, good evening, everybody. This is the International Academy for Clinical Hematology around the globe and across the hematology field. I'm Mohamed Moti from the Sorbonne University and Saint Antoine Hospital in Paris, in France, and it is my great pleasure to welcome you to this highly anticipated webinar dedicated to the latest advances in myeloproliferative neoplasms, or MPNs, as presented at the last American Society of Hematology 2024 annual Congress that was held in the beautiful city of San Diego. So we are thrilled to have such a diverse and engaged audience, especially on a Friday, from around the globe, united by our shared commitment to advancing care and outcomes for patients living with MPNs. Today, we have the honor of hearing from one of the foremost experts in the field, namely Professor Naveen Pemmaraju. As many of you know, Professor Pemmaraju has dedicated his career to pushing the boundaries of research and treatment of MPNs. His work has not only transformed our understanding of these diseases, but also paved the way for innovative therapies that are making a tangible difference in patient lives. So in the session, Professor Pemmaraju will provide an insightful overview of the groundbreaking findings presented at this year's ASH Congress, from novel treatment approaches to evolving strategies in disease management. He will dive into the latest data and explore its implications for our routine clinical practice. So before we begin, I'd like to remind everyone that there will be a Q&A, a live Q&A segment at the end of the presentation. So please feel free to submit your questions through the chat box during the talk or at the end, and we will address as many as possible. So once again, thank you for joining us. Let's make this an engaging and inspiring session. So without further ado, it's my great pleasure to welcome Professor Pemmaraju. Naveen, the virtual floor is yours.
Thank you, Professor Moti, for this not only beautiful and warm introduction, but highly inspirational. My love for you, for Professor Nagler, for your entire team at the IACH, International Academy for Clinical Hematology, continues to expand and grow. You as my mentor, friend, collaborator, close personal confidant and advisor, thank you so much for this platform, and thanks for all that you do before, during, and after COVID-19 pandemic to continue to connect all of us, particularly in these rare hematologic diseases. Thank you, Professor Moti. So my friends and colleagues, I see some of you logging in. Let's spend a half an hour together going over updates in the MPNs, just fresh off of the ASH hematology meeting. And so to set the landscape for the myeloproliferative neoplasms, or MPNs, as we enter into 2025, I'd like to show you this slide to show you all the progress actually in just the last few years. So remember, as we talk about MPNs, the three classical ones, polycythemia vera, or PV, there was an FDA approval in both Europe actually and US for ropeg interferon 2 to 3 years ago. That's being used in the clinics. And there are several agents either in phase 3 testing or finishing, including the rusfertide PTG300 hepcidin mimetic, which we presented an update by myself and Professor Aaron Gerds. In essential thrombocytosis, or ET, I'm proud to tell you there are several phase 2 and phase 3 trials going on, both in the interferon class as well as LSD1 inhibitors. And then in the rare MPNs, systemic mastocytosis, several approvals focusing around the KIT mutation, with several others in active clinical trials. And then finally, the rarest of the rare, ultra rare, 8p11 FGFR MLN syndromes, one approved drug already, Pemigatinib, and I'll show you a slide on a new agent that's being actively tested in this rare disease field. But of course, most of our field now is dominated, appropriately so, by clinical trials in myelofibrosis. And I'd like to show you those updates from the ASH meeting slides provided by my colleagues and friends. Remember, there are 4 approved JAK inhibitors, at least in the United States, which are ruxolitinib, first in class, as many of you know, based on the COMFORT 1 and 2 studies, followed by Fedratinib, approved in 2019, pacritinib in 2022, and momelotinib just last year. The excitement in the field centers around the combinations, and I'll show you the latest phase 2 and phase 3 data. And then, of course, moving beyond JAK inhibitors into the completely novel agents, those that help anemia, and those that function in other pathways that we have never thought of before in the MPNs. So with that landscape, we know that the first-in-class ruxolitinib has been approved now in many areas for over a decade plus. This JAK1, JAK2 inhibitor really revolutionized the field. Although, however, it does not necessarily cure the disease as a monotherapy, as oral pill chemotherapy. In this profound study by Professor Palandri from Italy, strikingly at 3 years, almost half of the patients stopped taking their JAK inhibitor, mostly due to adverse reasons such as loss of spleen, AEs, so adverse events, progression of the disease, infections. Only a few of these patients came off for a good reason, such as allogeneic transplant. The readout from this is that the median overall survival after the Ruxo is discontinued or is taken off is only around a year. And the only major factor that improved the overall survival, of course, outside of going to allogeneic transplant, which is curative for these patients, is investigational agents or access to clinical trials. I asked the question a few years ago, and now the field is really nicely debating this topic, and it's about disease modification. We have a set of pills, the JAK inhibitors, that which do not cure the disease, but certainly can put it into a remission state, can palliate the patient, can even improve the overall survival in some cases. But again, we're not curing the large majority of these patients without an allogeneic stem cell transplant. And so with this paradigm in mind, before I show you the latest from ASH, I would put forward 3 factors. 1, can we start to treat patients earlier before they develop overt advanced MF? 2, can we combine drugs beyond JAK inhibitor? And 3, once the JAK inhibitors completely stop working, are there novel agents that we can introduce, as our colleagues in myeloma, AML, MDS, and others are exploring? Well, I'm happy to show you that the menu, if you will, for novel agents continues to increase essentially weekly, with collaborators around the world, Europe, Asia, everywhere, really contributing, South America, to this new clinical trial landscape. Essentially, these drugs can be divided by the traditional models of extracellular, intracellular, nuclear, but also these new pathways such as telomerase inhibitor, CALR mutational directed, JAK2 mutational. So the mutation-specific era and immunotherapy era now begins in the MPNs. So without further ado, let me sample some of the studies that I thought were the most groundbreaking studies. The first study I'd like to highlight is from my own colleague here at MD Anderson, Dr. Lucia Masarova, who is leading a very innovative study, which is a phase 1, so an early stage study of a novel approach called T regulatory cells, which I'll call Treg infusions, in patients with myelofibrosis who are on a JAK inhibitor, but not quite having the optimal response. And so this is add-on therapy. In this ASH oral presentation, the first 9 patients enrolled were highlighted. And again, what the concept here is inflammation. JAK inhibitors are an outstanding way to decrease the inflammation, but again, may not be curing the disease. And so the question is, are there other modalities? This is an allogeneic, so off-the-shelf, cord blood-derived regulatory T-cell program sponsored by the company of Selenkos, which adds this infusional product with very minimal side effects to patients already on the oral JAK inhibitor. And this sort of second study, building on the first study, features CXCR4-enriched Tregs, again, to try to decrease inflammation. The overall concept is that regulatory Treg cells appear to suppress inflammation and restore equilibrium. And this product is actually very cleverly made in that it does not require matching and has minimal side effects with a quick infusion time and can be done as an outpatient. In this phase 1b study, so far 9 patients have been enrolled with these infusions. and spleen volume has been measured in the first 6 patients. So obviously a very early study, preliminary but encouraging results. Already starting to see spleen volume reductions in several patients, which is one of the hallmarks for our field, as well as improvement in the early satiety, also a very big factor with these patients with large spleens. And there is a symptom reduction signal already being seen, possibly in the setting of the hypothesis of improvement in the inflammatory markers, so decrease in the inflammatory markers. And finally, one novel aspect of this Treg program of Dr. Masarova is the reduction of TGF-beta levels. This is a new focus, or an old and new again focus in the MPN field. And this reduction in this key inflammatory marker appears to correlate with clinical responses in the first few patients seen. And so this trial is ongoing, enrolling at our center at MD Anderson, and we hope to update these data in future studies. So that's a Treg inflammation immune pathway approach. The second study I'd like to highlight was presented by our friend and colleague, Professor Mascarenhas from New York, which is the results from the randomized multicenter international study, phase 3 study, so-called Boreas, which features a novel MDM2 inhibitor, so another new pathway. Navtemadlin is the drug, versus best available therapy. These are already patients who've had a JAK inhibitor and it was not working for them. Another new pathway, so MDM2, this one appears to be a very important factor in patients with MF. The key is that MDM2 overexpression appears to prevent TP53-driven apoptosis of these CD34 positive or stem celled MF cells. And in so doing, if an inhibitor to that pathway can be provided, perhaps decrease inflammation through this MDM2 pathway. So in this very nicely done phase 3 study, again, randomized study, patients should be TP53 wild type, so they should not have the TP53 mutation as the MDM2 signals through that pathway. And in this study, you can see a large study, 183 patients, relapsed refractory after a JAK inhibitor, 2-to-1 randomization, the new drug navtemadlin MDM2 inhibitor, 240 milligrams Q-day, days 1 through 7 on a 28-day cycle. That's 123 patients versus best available therapy, 60 patients. So 2-to-1 randomization. Standard endpoints of spleen and symptoms, but also I really applaud the investigators for including these biochemical endpoints, including looking at CD34 stem cells, cytokines, bone marrow fibrosis. So obviously looking to see for potential markers of disease modification. In this large study, SVR35 at week 24, keep in mind these are relapse refractory high-risk patients, as shown here, 15% SVR35 in the MDM2 inhibitor treated group versus only 5% in BAT. Looking at the TSS50, so that's our MPN total symptom score burden at week 24, very nicely shows the navtemadlin at 24% versus only 12% in BAT. This is showing the absolute change in the total symptom score at week 24. You can see by the waterfall plot quite a few patients benefiting in terms of their TSS. I mentioned looking at potential markers for disease modification. I think this should be a mandatory requirement for all of our larger studies in the MF field. And as you can see here, the investigators found that there was a driver gene variant allele fraction reduction, VAF reduction, and bone marrow fibrosis improvement, all quite early at week 24. VAF measurements by waterfall plot on the left, bone marrow fibrosis by central pathology review on the right. Again, we are trying to establish what disease modification looks like. These are some of the early candidates for that. How is the drug tolerated overall? A well-tolerated program. It's already known that the MDM2 inhibitor class as a whole will have GI side effects as highlighted here, and of course hematologic side effects as these are potent modulators of inflammation in MF, anemia, neutropenia, and thrombocytopenia with the numbers shown here. Overall, there were no new unexpected safety signals. This is a well-tolerated oral drug program. And so this Boreas trial concludes as a relapse refractory study by denoting not only improvements in spleen and symptoms in an otherwise very high-risk group of patients, but also markers of early disease modification. And there is an ongoing phase 3 frontline study with this MDM2 inhibitor now in progress. A second oral presentation, also by Dr. Mascarenhas and group, highlighted the same trial I showed you, but the biochemical correlatives and the disease-modifying effects from this phase 3 Boreas trial. And just a couple more slides to highlight that. I really appreciated this companion presentation, which showed more detail of what we just said. And so here you see that the CD34 positive and VF changes actually correlated with SVR. This is among one of the first times we've seen that in the field. where the navtemadlin and MDM2 inhibitor really reduced this level of circulating CD34 positive cells. And furthermore, they nicely showed reductions in inflammatory markers that correlate again with SVR. Again, the MDM2 inhibitor reducing pro-inflammatory markers, some of the common ones shown here. And again, I'll highlight this TGF-beta, which I showed you in the last study of the Masarova Treg study. And finally, as I mentioned, this navtemadlin MDM2 inhibitor program, based on these encouraging results, now has moved into the front line in the very exciting, large, and ambitious study, approximately 600 patients planned, so-called POESIS study, which will be a phase 3 randomized double-blind study, international, adding on the new drug navtemadlin to the JAK inhibitor Ruxo versus Ruxo and placebo, frontline JAK inhibitor naive patients with myelofibrosis who have a suboptimal response to ruxolitinib treatment. Of course, we'll be eagerly awaiting those results as they're updated over the coming year plus. A next study I'd like to show you is from Dr. Andrew Kirkendall of the Moffitt Group, who highlighted a very nice investigator-initiated study program of a phase 2 program of Fedratinib, the second approved JAK inhibitor, in patients with the rare disease of MDS-MPN and chronic neutrophilic leukemia. In this multi-center IST, the concept is that MDS-MPNs have been not only rare, but also challenging to treat, as they obviously share features of both MDS and MPNs. And actually, a lot of these patients will have overall survival problems and progression to AML. These patients have been heterogeneous previously, but defined in this study as CMML, atypical CML, MDS-MPN RST, and the MDS-MPNU are unclassifiable. There have been a few studies in this field, one with the JAK inhibitor ruxolitinib, which did appear to show benefit in a subset of patients, but there were clonal progressions still seen. The hypothesis here that the Fedratinib or second-in-class JAK inhibitor has other targets outside of the ruxolitinib and outside of the JAK-STAT pathway, which may include bromodomain, FLT3, and even suppression of c-MYC. The c-MYC is interesting because it's been implicated in both lymphoid and myeloid diseases, and the hypothesis is that perhaps by targeting the c-MYC pathway, there could be clinical benefit. In this phase 2 study, we included, the investigators included these rare diseases, so a total of 25 patients were included, and the Fedratinib was given at a standard dose, 400 milligrams PO daily, with sort of the standard assessments week 12, week 24. Overall, the Fedratinib showed consistently reduction in spleen volume and improvement in symptoms in these difficult-to-treat patients, with waterfall plots of spleen I'm showing you here on the left and on the right. Again, an SVR rate of 35.2% and a symptom response rate of 50%. How was the Fedratinib tolerated? Again, no new safety signals here. It's already well known that there are GI side effects, a black box FDA warning for encephalopathy, including watching for Wernicke's encephalopathy. You can see the AEs in greater than 10% of patients here on the left, as expected, GI mostly, and then grade 3 or higher AEs on the right, including CBC changes as well. So overall, from this encouraging multi-center IST, we praise the investigators for looking into a rare disease set and also biochemical incorporation of correlatives, nicely showing that this hypothesis of could Fedratinib decrease the MYC staining, and they are showing that early on. Of course, this study is ongoing, and we look forward to future updates here. A very important study to highlight is from our great friend and colleague, Professor Claire Harrison of the UK. In this exciting study, she showed hematological improvement and other clinical benefits of a novel agent known as elritercept as both a single agent and in combination with JAK inhibitor in patients in an ongoing study, phase 2 for MF, so-called RESTORE trial. This is a very interesting agent. So elritercept, aka CARE-050, was really created in mind to target disorders of ineffective hematopoiesis, which includes our myelofibrosis. This clever agent, as shown here, is really designed to inhibit select TGF-beta superfamily ligands. So again, TGF-beta, very important, including activin A, which has been associated with not only ineffective hematopoiesis, but also increased inflammation, disease pathogenesis, and progression. These prior drugs do include luspatercept, sotatercept, other agents in this TGF-beta greater superfamily. So that's where some of you have heard of this pathway. I think the key here is we wanted to see if there were markers of disease modification beyond anemia improvement, which would have been hypothesized from first principles. In this large study, You have multiple cohorts, again, monotherapy, so the elritercept itself, part 1 dose escalation, 0.75 mg per kg up to 4.5 mg per kg, and then a part 2 dose expansion, again, monotherapy and in combination with ruxolitinib. Overall, 73 patients included in this safety analysis, 20 for the spleen response evaluable, 27 for the symptom response evaluable, and several of the patients included in the anemia improvement. Overall, in addition to the expected or hypothesized, I should say, hemoglobin responses, which were observed with the elritercept treatment, as you can see here, and again, this is both arms showing hemoglobin improvement, including transfusion burden decrease. I think also very nicely shown by Professor Harrison was reductions in spleen size that were observed with the elritercept treatment. Again, this is both monotherapy and combination arms. So starting to see markers potentially early on in the study again of disease modification beyond the anemia in the spleen. Importantly, overall in both arms, reductions in spleen size were observed in the evaluable patients already by week 24, 8 out of 20, so that's 40%, had a reduction of 10% or more, and 15% had a reduction of the more gold standard, 35% or more. So we await further results of that program, which is also going to be put forward in multiple other myeloid malignancies in other states, such as MDS. A few other studies to highlight, if the viewers will allow, my own study featuring our superstar hem-onc fellow, Dr. Hannah Goulart. This was something that's a very important, dear topic to me that I've been writing and thinking about for over a decade, which is adolescents and young adults with MPNs, so-called AYA. We looked at our MD Anderson series along with my colleagues, Dr. Lucia Matsarova, Dr. Bose, and our chairman, Dr. Kantarjian. And in this study, showed a couple of new findings in this update. AYA patients made up approximately 454 patients as compared to 3,000 plus in the older group. We defined AYA as up to age 40, so below age of 40, 15 to 39, and try to look at ET, PV, and MF. I think if you look at the Kaplan-Meier curves, very telling story that there are patients who do transform to AML, to myelofibrosis from PVET to AML, and that the overall survival in AYA patients is not 100% in all patients. So young patients do get MPNs as well. They can have transformation events, and unfortunately, a subgroup of patients who may unfortunately experience early death. We did find now in the molecular era that many of these patients have high molecular risk mutation analysis, and we did find that patients who appear to have an HMR mutation may potentially have a worse outcome. And so maybe one readout from this is in our young patients, we need to be thinking about, are there potentials for early disease modification, interferon or other agents if no contraindications, checking for these high-risk molecular mutations beyond JAK, MPL, and CALR, and then really evaluating patients early on for curative therapy, such as allogeneic stem cell transplant, if feasible. One more study to highlight is that of the rare disease I mentioned earlier in the opening remarks, the FGFR-expressing MLN neoplasms. And interestingly, we have one drug already approved, Pemigatinib, the FGFR inhibitor, which is also approved for cholangiocarcinoma in the US. But there's a second drug being actively investigated, that of olverembatinib. Again, in this rare disease, olverembatinib, which is a multi-kinase inhibitor, has activity against FGFR1. And so this drug has already started testing early on, but in chronic phase patients with the FGFR MLN, all 7 patients appear to have had a CR, a major response, with 5 alive and several patients going to transplant. There were also patients treated with more accelerated blast phase as well. And so we look forward to seeing further results in this rare disease field. I think I would close these remarks to say a few things on the last 3 slides. One is I showed this slide earlier, so same slide. Again, just looking at the novel agents, there was again another nice talk by Professor Mascarenhas on the imetelstat, which is the first-in-class telomerase inhibitor. He's combining that with the ruxolitinib in an early phase study. First 17 patients reported an oral presentation showing not only safety but efficacy, especially at the 9.4 target dosing. So that study is ongoing. We mentioned LSD1 inhibitors. ASH last year, I on purpose didn't show new slides here, were the BET inhibitors and BCL-XL navitoclax, both of which had positive phase 3 studies presented last year. I presented navitoclax, Dr. Rumpal pelabresib. But amazingly, to remind the viewers, even though both of those met the spleen size, they both 60+% for the SVR35, doubling what was in the control group, Ruxo plus placebo. Both of them just barely missed the p-value of significance for symptom benefit. And so at this current time, neither of those drugs are approved. So positive primary endpoint in phase 3, p-value not significant, so the symptom burden was not met. So neither of those drugs currently approved. So we'll have to wait to see what are the future development plans there. I mentioned the two most active current phase 3s, the MDM2 inhibitor, navtemadlin, and I also should mention selinexor, which is an XPO1 inhibitor, also currently in active phase 3 frontline randomized combination. So we eagerly await that. I think the key for our field, Professor Moti and colleagues, will be, can we reimagine the field beyond spleen and symptoms, which served us well prior to no therapies and in the JAK inhibitor monotherapy era? Now, as you're seeing a host of different drugs, novel agents, combinations, mutation-specific drugs coming, CALR and JAK2, immunotherapies, just as has been done in solid tumors, in AML, MDS, and multiple myeloma. Can we develop an MRD endpoint, which we never have? Can we focus on overall survival, PFS and EFS, which we have not traditionally done? And can we focus on getting patients to allogeneic stem cell transplant and the accelerated blast phase, decreasing the blast count? I think finally, what we need to do is move towards a greater emphasis on disease modification. Again, I would say overall survival being the main readout. I would also preview folks to say very exciting now in phase 1, just hitting the clinic around the world, is mutant-specific or type 2 specific JAK2 inhibitors. And then CALR can be targeted through various immune properties such as bispecifics and monoclonal antibody, which has already entered the clinic. And then Alex Romputos and colleagues showed exciting data on the beginnings of preclinical work against CALR for CAR T cells. And with that, I'll pause. I'd like to turn it back to Professor Moti for some Q&A and some discussion. Professor Moti?
Thank you so much, Naveen, for really an amazing summary. And obviously, like any summary, you need to make choices, but I think you selected the best of the best. And we already have a lot of questions, and we won't be able to take all of them because of time. And I know some people are rushing now to start the holidays, at least in Europe. It's becoming late afternoon. So one question is about PV. What would be, in your opinion, the first-line therapy of PV today? What is the algorithm you would adopt? And I guess the issue is about whether hydroxyurea is still first line or whether we should move to interferon alpha.
Wow, great question. So two things for PV. One, I should mention Professor Teferi's beautiful oral presentation showing that even in PV, the earlier MPNs, if you have a multi-hit TP53, that appears to give worse outcomes. So that was a nice study in that field. Now, for the clinical question that you nicely asked, PV for 2025, I would say three things. One, we are still doing low risk versus high risk. Low risk is usually defined as less than age 60, no prior blood clot. High risk is one of those. If you're low risk, the standard of care remains baby aspirin, if no contraindications, and phlebotomy for hematocrit goal less than 45. But as you're asking about really high risk PV, we call it triple therapy. So in addition to the baby aspirin and the phlebotomy, hematocrit less than 45, you want to give a cytoreductive agent. In the States, your choices are hydroxyurea, longstanding, or I favor an interferon-based product if there's no contraindications. You have the Pegasus or pegylated interferon available, or now the Besremi or Ropeg interferon, which has been approved in the States since 2021. So if no contraindications, I and others favor an interferon-based. There are studies, two studies head-to-head, they're still ongoing, of Hydrea versus interferon. Professor Moti, my guess is it may not answer that debate. I think there'll be patients who will need one or the other. And we should also mention there's the ruxolitinib, the JAK inhibitor, which is approved in the US for after hydroxyurea intolerance or failure. That is also an FDA-approved option.
Excellent. Thank you very much. Same question about ET. What is the current standard of care?
ET, I have to say, as you guys saw from the slides, not a whole lot of development historically, but there is now. So standard of care, again, low risk, high risk. kind of the same paradigm. If you're under age 60 or no prior clot, you can do baby aspirin alone. There is a movement in the field that you can be so low risk that you can just be observed alone. Then in the high risk, double therapy here because there's no role for phlebotomy. So again, hydroxyurea versus interferon-based product and baby aspirin. The major movement in the field is, can there be something to replace? So LSD1 inhibitor has moved momentumstat to the frontline randomized phase 3. So that's a potential registrational strategy. BET inhibitor appears to have activity in ET as an active investigation. And again, the ROPEG interferon, which only has the approval for PV. So my prediction for us is in the next 3 years, we'll have a one or two new active agents and options for ET, but not yet.
Beautiful. Thank you so much. So now I come to the dozens of questions about myelofibrosis because there is such a high level of excitement about myelofibrosis. And I must confess, this disease has been moving, I would say, in the right direction now.
Yeah.
With many great drugs. You highlighted ruxolitinib. We have a few questions about momelotinib. So can you, the question is about the practical use of this drug. What are the best practices in your practice? Where does this drug fit? And what are, I would say, the precautions one would need to bear in mind, or maybe it's it's very easy to use. So can you
Sure.
just highlight this to the audience?
Yeah, I'm glad this was asked, Professor Moti, because this is a good one. This is our 4th approved JAK inhibitor, only approved in the US September of just 2023. In addition to JAK, it also hits a pathway that's very important called ACVR1. And we think that that's in part why it actually got approved, which is MF with anemia. That's the actual on-label indication. It was finally put over the finish line by the Momentum study, which was a randomized phase 3. Interestingly, prior JAK inhibitor, 2-to-1 randomization, the momelotinib agent, oral drug, JAK inhibitor, versus danazol, and it did show not only improvement in anemia, but also spleen and symptoms. So we're using it in the US either as a frontline agent for someone with anemia, or probably most people are using it as a second agent or beyond after Ruxo or other frontline therapy. I would say 3 practical measurements that we're seeing in the clinic. One is that it can have a signal for peripheral neuropathy that was shown by Professor Teferi and others in the early studies, so you should be looking out for that. 2, there is some signal that it can affect LFTs or creatinine, so liver kidney function, so that's important beyond the normal CBC. And 3, some patients can get myelosuppressed with the agent, so they can have anemia and thrombocytopenia before those numbers actually improve. So this is one of those agents that I am checking blood counts regularly in the first few months after prescribing it, seeing patients back. But overall, it could be a game changer, Professor Moti, for patients with MF and anemia, which has been this sort of unmet need. We've been either giving the luspatercept or EPO, blood transfusions, danazol. So I think it can represent that. And then there are still open studies to combine the momelotinib with other agents too, right? So with BET inhibitors, BCLXL, luspatercept, all of that. So it's really approved for MF with anemia, and it opens up that new door.
So in the same line, we have a question about the use of momelotinib.
Oh, good.
Before transplant and after transplant, because obviously I don't know, unfortunately, I think many patients, whenever possible, will still need allotransplant in MF. And if they have started on momelotinib, are there any rules one needs to follow? And can we actually use it after? Because we know that allotransplant will not cure MF immediately. It's a long-term process.
Right. Well, great question. For the only definitive part of the question that we know, as you said nicely, I want to reemphasize for our viewers, allotransplant is the only curative modality for myelofibrosis. These drugs and combinations are still being tested for that. So that's number one. 2, as you said, this is all what's actively being studied. We've done a lot of those studies in ruxolitinib, the first JAK inhibitor, but those studies are just now happening for pacritinib and momelotinib, the newer JAK inhibitors. So you asked 3 important questions. So at least in our own clinic experience, anecdotally, we know it's safe to give momelotinib up until the time of transplant. Folks will need to work out tapering and all of these things as we did for Rux. Two, there's no standard of care for momelotinib post-transplant, but people are studying PAC, momelotinib, Rux, fedratinib post-transplant. And then three, I love what you said, which is unfortunately, people can still not respond or relapse post-transplant. So we are starting to get those patients in our clinic. So not as maintenance yet because we haven't studied it, but I can tell you that we have treated patients post-transplant with JAK inhibitors, and we know that that's feasible. So basically a lot of active areas of investigation you brought up there.
Wonderful. Thank you so much. Let's move on to another series of questions about actually selinexor. I must confess, I was very surprised to see you guys in MPN bringing selinexor into the forefront because obviously selinexor is approved and relatively widely used now in relapsed multiple myeloma. It's an XP1 inhibitor. And then I thought, but how come it works in MPN? And my spontaneous thought was, oh, myeloma is MYE and myelofibrosis MYE. So there is the three letters in common, so we can immediately jump in. Is this the scientific rationale for
That's
doing
as good as any explanation I've ever heard, Professor Moti. Yeah, so it's worth talking about, especially with your expertise in myeloma. This is one of the surprising clinical developments of the last few years. To bring viewers up to speed, anyone who's watched your programs, the selinexor ultimately got approved in myeloma and lymphoma, but with two major caveats. The dose is a bit higher than what we're using here, and also there's a real signal for nausea, vomiting, GI side effects, fatigue. So here we've brought the dose down. So I'm going to tell you it's as low as 40 Q weekly or 60 Q weekly. So that's how low the dose is now. So markers of magnitude lower than what you guys study myeloma. And two, unfortunately, the nausea, vomiting, GI signal is still there, but we're mandating folks to take even up to 2 anti-nausea medicines on this trial. So what happened was this XPO1 pathway, as you said nicely, appears to be active in multiple different myeloid, lymphoid, so again, lymphoma, myeloma, myelofibrosis. I think beyond the XPO1, it probably has anti-inflammatory activities as shown in their cytokine panel. So early on in a small pilot study, first 14 patients, some of the highest SVR35 reductions that we've seen in the field, in addition to symptom benefit, despite the GI signal. And so based on that excitement, it has gone quickly and I think very nicely into a randomized phase 3. So it'll be Ruxo selinexor versus Ruxo plus placebo. 300 plus patients expected, symptoms and spleen. So I think this is very important. Can you show symptom benefit even with this known GI nausea vomiting? I think so, because you have a reduced dose, you're paying attention, what we learned from you. And then I think the mechanism of action still needs more understanding. I think this is a multi-pathway influencing anti-inflammatory drug that needs more science, and they're doing those studies right now.
Excellent. Thank you so much. And this is really very promising and very exciting because we will probably need all of these drugs. Maybe I'll take one last question for the last minute. And this is a more global question because it's based on your answer a few minutes ago saying we have 4 approvals of drugs in myelofibrosis, different molecules, different mechanism of action, which is really great, plus allo transplant. So treatment options are becoming quite diverse.
Right.
How do you sequence these different treatments? And would you envision that a given patient would maybe need all of them at the same,
Yes.
What is a patient experience, I would say, or patient journey, if
Yeah.
you prefer?
That's great. No, I think this is an awesome question to end on. So yes to everything you said. NCCN guidelines and other guidelines are attempting to answer that question. But as we all know in the clinic, sometimes it's a bit more heterogeneous and more personalized. In general, with these 4 JAK inhibitors, there's no molecular necessarily guide. So it is clinical factors. So again, each one's approved in a slightly different way. Ruxo and Fedratinib really approved more on the traditional spleen and symptom reduction. Both have broad approvals. You can use them frontline or later. But what's interesting is they cut off at a platelets of, it should be 50 or above. So these next two JAK inhibitors, pacritinib and momelotinib, were actually smartly designed with that in mind, and so you can deliver them in the setting of cytopenic MF. So that's become the biggest branch point in clinic for frontline therapy. Platelets, 50 or above, yes or no. If below 50, then you have the pacritinib. If anemia or platelets drop, you have the momelotinib. And then, of course, you always have low-dose ruxolitinib and frontline options. If you can, a lot of us favor giving the newer agents because you can deliver a higher dose rather than reducing the prior. Then, as you said, number two is, yes, I do. Like in CML, perhaps if you'll allow in myeloma, now that we have these drugs available, right, the natural history is being altered. Patients are living longer and better. And because these are oral drugs, easy to give, and I hope that they'll be available worldwide soon, yes, I see a sequencing. And maybe if you'll allow, maybe we will also see biological reasons to choose these. So selective pressure mutations, resistance mutations, we haven't seen that yet. And then finally, the combinations. I hope that the combinations will emerge as some sort of a guidance and say, this combination is superior to all these monotherapies, and either use it as an induction followed by maintenance, or that we have other novel agents in there. So I think that's kind of it. And then again, let's get people to transplant who qualify. Let's look for donors. Let's start that process early, and let's dynamically reassess transplant before and after each JAK inhibitor.
Wonderful. Thank you so much. As we conclude, I'd like to emphasize the importance of translating these innovations into meaningful outcomes for our patient. I hope today's discussion will be the catalyst for further dialogue, collaboration, and action in advancing this field. Thank you all for joining us today for this insightful webinar on the latest advances in MPN presented at the last ASH meeting. We had the privilege of delving into cutting-edge research and clinical updates by the very well-known Professor Pemmaraju. His expertise has not only illuminated key development, but also inspired us to think critically about how we can implement these new findings in our practice. On behalf of the International Academy for Clinical Hematology and all of us here, I extend our deepest gratitude to you, Naveen, for sharing your expertise, and to each of you for your active participation. So let's continue to stay connected, to stay curious, and remain committed to advance knowledge in MPN. And as the holiday season approaches, I want to take a moment to thank you all for being part of our journey this year. Your dedication, collaboration, and passion inspire us to keep striving for excellence. So may the season bring you joy, peace, and some lovely cherished moments with your loved ones. Wishing you happy holidays and a new year filled with health, success, and new opportunities. So we'll take a break for the next 10, 12 days. You'll have a couple of 2 podcasts, but the educational activities of the IACH will start again on the 6th of January with the new initiatives, Pioneers in Hematology. So please stay tuned, and thank you all, and wish you some happy holidays. Thank you very much.